and mechanical properties of the cells and areas in general. So, okay, so as you guys may have kind of seen that I was hinting at, it's kind of a false dichotomy to try to separate mechanics from biology in these systems, and there are a few reasons for this. One is when you change the behavior of a cell or change the gene expression of it. So this is like what proteins the cell is making. This will actually affect the mechanics of the cell, right? And so one example of this is if the cell increases its production of myosin, which is a protein that acts as a molecular motor inside the cell. This will increase the forces that the cell generates. And then we also see that sort of the reverse is also true. So if the environment of the cell changes, so say the area around the cell is more stiff, the forces exerted by the cell will also change in response to this, right? And so the sort of the study of how the mechanics of the environment affect the actual biological behavior of cells and tissues is actually a field called Mechanobiology, which is a very exciting field that's seeing a lot of developments happening right now. And to show an example of this mechanobiology in action, I'll just use kind of a very classic example here. Here I want you guys to focus on understanding sort of this plot right here, right? And so what we have going on is we have a cell that is sitting on top of a gel, right? And the scientists who did this experiment gradually, well, they use gels of increasing stiffness. And so here we can see that this gel here is 1 kilopascal, whereas the gel all the way on the right is 10 kilopascals, right? And so we see that as the scientists increase the stiffness of the gel, the force, here this is the force in micronewtons exerted by the cell, the force increases almost linearly. And this also changes the actual area of the cell. And then for our reference, they also included plot A divided by plot B. And so this is just one of many examples of how changing the mechanics of the environment can affect how a cell actually behaves. Okay, so I'm just going to look at the questions real quick before I do one more topic. Okay, so here I'm seeing a question here. Can you elaborate on how the information of these morphological changes is encoding at different levels of life, if not at a genomic level? Okay, so I see now that Josh would like to answer this question live. So,
no, I just meant that you were answering it live. Go ahead.
Okay, great, thanks. Great. As mentioned by this question, clearly, mechanical changes are present in the genome, and our DNA governs how our cells behave. But I think kind of the maybe more important process going on around these types of things is actually gene expression. And so if you guys may not know what gene expression is, so this is say if you have two genomes that are exactly the same, the cells with those genomes may behave differently, because the cell will sense its environment and then choose to sort of produce proteins on certain genes and not produce proteins on other genes. And so in this way, the mechanical environment of a cell can very directly change and influence how a cell behaves, and how it interacts with its mechanical environment. Okay, so I'm going to move over to my last slide here that answers another section of questions here. And so this question, we got several questions around how do we study how kind of the biology of our system actually directly interacts and changes the mechanics of our system. And so I'll talk about how we do this. So we sort of need to do two things to be able to connect molecular and genetic mechanisms with the actual mechanics of our cells or our tissues or our organisms. And so the first step is altering the behavior of the cell. And so there are a couple ways scientists do this. One is using certain kinds of drugs. So you may use what's called an inhibitor to reduce the gene expression of a certain gene, and so then you would make less of a certain protein. Right? Another option is to sort of alter the organism's genome directly, and so you'll see this done pretty often in Drosophila experiments and studies where they will remove a gene and then see how that affects the process. And so that brings me to step two, where after you've altered either the cell or the organism, then you observe what happens to it. And so the claims will usually fall in this structure. So if the change using the inhibitor sort of destroys the process, then a claim we could make would be a certain gene or protein is necessary for a certain biological process. So gene or protein X is necessary for biological process Y. Right. And so one challenge with this approach is that when you change a protein or you change a gene in a cell or an organism, this will affect many things in the organism or many things in the cell, and so it can be difficult to tease apart exactly which change is affecting which specific process, and so that's why the structure of the claims looks like what I mentioned there. And I'll just show one quick example of this from a paper here. And so the claim here is that the gene Dizzy or the proteins Dizzy and RAP1 are necessary for ventral furrow formation. And so this is going back to the Drosophila gastrulation process that I talked about earlier. So I'm just gonna spotlight this. And so you usually see these experiments with first a wild type, and so this is what we would expect in nature, and this is sort of the healthy example. And then you'll see other columns where certain proteins are either greatly reduced or completely eliminated, right? And so this Dizzy column doesn't mean that they added dizzy. It means that they actually took away dizzy. And then here the same things for RAP1. Right, and so what we see here is that normal Drosophila development goes as it should, and then in the case when they knock down dizzy, we see that it isn't able to completely close down here, and that we're not forming the fold here. So we see a fold happening here that isn't forming here, and then this is even more exaggerated in the case of RAP1. And so I'll take a peek at our questions again. Okay, so I might do a quick time check. So I'll answer this first question and then I'll go ahead and pass it on. Okay, so this question is actually on the previous, so let me answer this one live. This question is on the previous section, talking about how cell area was affected by the stiffness. Right, and so does the area covered by the cell decrease as the force exerted increases? So this is a good question. I think there's just a little bit of confusion as to what we mean by the cell force. Here in the picture, right, we see that when the cell exerts a force on the substrate, so when the cell contracts, we see the cell decreases in size, right? And so I think it's very reasonable to think that if the cell is increasing its force, then the cell would get even smaller, right? But what actually happens when a cell spreads is that as the cell pulls, it actually flattens, and so that's why its area increases. The exertion of the cell force and the area of the cell are actually two independent processes here. And so we don't know for sure that if the cell force is increased, whether the cell area will increase or decrease. Here we have both of these things measured, and we're seeing that in general, the pattern goes that cells that are spreading more, so as we see the area increasing on these cells, these also correspond to cells that are exerting higher forces. I may compare this to something like if we stretched, say, a spring, or something like that. As you stretch the spring more and more, you may leave sort of the linear or soft regime, which is something Sijie will talk about. And so as you stretch the spring more, it may actually pull back harder. And so in the case of something like a cell, where if the cell was stretched so much that it fell apart, that'll be, of course, very bad, right? And so there's an incentive for the cell to sort of pull back harder and harder as it gets more and more stretched. So thanks for that question. In the interest of time, we'll go ahead and move on to our next section. But thanks for everyone for your attention and questions. Okay, so I've stopped screen sharing. Sijie.
Okay, so can you see my screen? Yes, so many of you were asking about these new Hookean models that are used in both of these two papers, and also the concepts of elasticity. So I will just briefly go through the basics of mechanics and help you understand these mathematical models and elasticity. So we use mechanics to, describe how the material is going to be deformed for a given applied load. So we need to have a description of the deformation of the material, the applied load, and how the applied load is related to the deformation. So let's look at this first example where this material element is under a uniaxial tension. That means the applied load is along one dimension only. And for this undeformed material element under uniaxial tension, it gets elongated along the z direction due to the application of this force. And we define this normal stress as the force divided by the area where the force is applied, and this normal strain as this elongation along the z direction divided by the original length of this material element along the z direction. And so for if the deformation is small, we have this Hooke's law, just like what you see for the Hooke spring. And this Hooke's law says the normal stress is proportional to this normal strain, and these coefficients called Young's modulus. So for the Hooke's law to be valid, we need to have small deformation, which just means this normal strain, which is a dimensionless variable, has to be much smaller than 1. And we actually kind of exaggerate the deformation in this schematic diagram so you can see this elongation. But actually for Hooke's law to be valid, this deformation is almost like invisible. And also, of course, the material has to be elastic, which means if we release the applied load, then the material should recover the undeformed configuration. And this Young's modulus just is to measure the stiffness of the material. And if you have higher Young's modulus, the material have smaller deformation for a given loading condition. So there are other forms of deformation. One is for the material under this hydrostatic stress or hydrostatic pressure. And we have similar Hooke's law, which says this relative volume change is proportional to the hydrostatic stress. And this coefficient K is called bulk modulus. And so for a material under a hydrostatic pressure, it will only change the volume, but the shape will not change. That means any angle between any two infinitesimal line segments will not be changed due to the application of hydrostatic pressure. And so for this cubic material element under hydrostatic pressure, it would just be compressed to be a smaller cube. So another form of deformation is this shear. So under shear force, the material element will change its shape, but the volume is conserved. And we define the shear stress as this shear force divided by this area where the force is applied. And we call the angle change shear strain. So if the deformation is small, we have these approximation for the shear strain, which is just this delta divided by LZ. And we have a similar Hooke's law, which says the shear stress is proportional to the shear strain, and this G is called shear modulus. So all these shear modulus, Young's modulus, and bulk modulus, they describe the stiffness of the material. And for most biological tissues, the stiffness of them actually is so the stiffness is actually pretty small compared to these non-living materials that you see in your daily lives, like ceramics or metals. So for ceramics or metals, their stiffness is on the order of like gigapascal. But for these biological tissues, the stiffness is on the order of kilopascal. And in this figure at the bottom, I'm just showing for brain tissues from different brain regions, what's the shear shear as a function of, the shear stress as a function of the applied shear strength. And as you can see, these brain tissues can experience large deformation, and these stress strain curve actually are pretty nonlinear. And so these linear elastic theory cannot actually apply to study the mechanical properties of biological tissues. So we need to resort to some kind of nonlinear theory, and this new Hawking model is just one of the simplest nonlinear theories we can use. And to understand these new Hawking models, we need to use this finite deformation theory. So in the linear elastic theory, the undeformed configuration overlaps with the deformed configuration because the deformation is small. And in the finite deformation theory, since the deformation is finite, we have this mapping from initial or undeformed configuration to the deformed or the current configuration. So this mapping phi basically maps every point, capital X, in the undeformed configuration to the corresponding point in this deformed configuration. And locally, we have this deformation gradient F, which is like a second order tensor, and it specifies the local deformation. So the meaning of it is just like a linear mapping, which maps infinitesimal line segment from the undeformed configuration to the corresponding line segment in the deformed configuration. And we have this expression at the bottom here to express this linear mapping. And basically, this deformation gradient F will contain the information about locally how the infinitesimal line segment is going to be elongated or compressed, and how the angle between any two infinitesimal line segment is going to be changed under this deformation. And for more information, you can go to this book to know more details about the theory of continuum mechanics. So for to model the biological tissues which are also growing, we need some extra ingredients. So we assume from our initial stress-free state, due to the growth, locally every material element will be mapped to this this virtual intermediate state, which is also stress-free. And this mapping is described by this linear mapping G. And from this intermediate stress-free state, we also have this linear mapping F, which maps to the current configuration or the deformed configuration, which could also experience some external load. And we assume that this total deformation gradient A have this multiplicated decomposition. So it just says this total deformation gradient can be decomposed to the growth part and this elastic part. And in New-Hawkian models, we have this string energy function, and it's a function of this second order tensor F, which, like I said, it describes the elastic part of the deformation. And this J is the determinant of this tensor F, which is just like a Jacobian from this intermediate virtual stress-free state to the current state. And as you have learned maybe from calculus, this Jacobian just tells us how locally how the volume element is going to be changed. And so if you take to the linear order, or if you take the small string limit, you can show that these New-Hawkian materials or New-Hawkian models actually is the same as this linear elastic theory. And in this figure at the bottom, I'm just showing for incompressible Newtonian and the linear elastic material under a uniaxial tension, the stress as a function of the strength. So at the small strain limit, these two curves, they actually coincide. But as you increase the strain, this Hooke's law, which basically gives this linear line in this stress-strain relation. But for Newtonian material, you have this nonlinear behavior for the stress-strain relation. And some of you were also asking about the assumption and the limitation of these Newtonian materials. So first of all, to use Newtonian materials, we assume the materials are isotropic, and for many materials are like many biological tissues, they are anisotropic. And to incorporate the anisotropy, we might need to use some other models. And also many biological tissues are actually viscoelastic, which means the materials have both the elastic and viscous characteristics. So it can dissipate energy and will have some relaxation process. And in these two papers you have read, these authors basically, they only focus on the equilibrium state of the system just by minimizing this energy function. And basically the assumption is the time scale in this growth process is much longer than the time scale in the viscoelastic process. And that means if you look at this growth process, the system is always at the equilibrium. And also, since we are using this continuum modeling, we assume the characteristic length scale of the phenomenon we care about is much longer than the length scale of a single cell. And for this wrinkling phenomena, the characteristic length scale will just be the wavelengths of the wrinkles. And for if you want to know more about the mathematical modeling of biological growth, you can use this book as a reference. And I think this is the end of my slides, and I will answer some questions you have along the way. So someone was asking, are we still assuming the F is this deformation gradient F is a linear map that would map straight lines to straight lines, but that would disallow any curvature from forming under the stress. So so this deformation gradient actually is a linear mapping which is defined locally. So it actually maps any, so you need to think of like any infinitesimal lines elements which under this linear mapping deformation, that this deformation gradient will be mapped to the corresponding line segment at the current or the deformed configuration. But if you zoom out, you can still, have the curvature form. Okay, so I think I'll just pass to Anvitha, and we can answer more questions afterwards.
Yeah, so today I'll be talking about how mechanical instabilities cause biological patterns. So first we'll try to look at what are these mechanical instabilities, and then we'll try to develop, how these occur in biological systems, and as a result, how patterns are formed. So we initially consider a toy problem to study the two kinds of equilibrium. So what I'm considering here is a ball contained in a paraboloid. So so in the case that so here the paraboloid is determined by a constant c greater than 0, and the energy of the system is given by pi, which is like just the potential energy due to the gravity, and this can be written as some the constant c into u square. So depending on whether c is positive or negative, we can see that the ball is in a stable or unstable equilibrium. So what do we mean by stable or unstable equilibrium? So in both the cases, we can see that the net force on the ball is 0. So then the ball is in equilibrium. But in case of stable equilibrium, if we perturb the ball from its current position, then it comes back to its initial position. But in the case of an unstable equilibrium, on perturbation, like the ball doesn't return to its initial position. So we can also see that the stable and unstable equilibrium coincide with like the minima and the maxima of energy. So this can just using this toy problem, we can see that mechanical systems always try to achieve a state of minimum energy. So and now extending this to the buckling of a beam. So when you have a beam which is loaded slowly, so then we can see that it snaps from a straight configuration and into a buckle configuration. So what happens here is that the energy profile changes. So before the critical loading is achieved, you have the energy profile which looks like this, where the minimum of the energy occurs for a flat beam. But when the loading is increased beyond critical loading, then the flat beam becomes an unstable equilibrium. So and it becomes the maxima. It becomes a local maxima. So what the system tries to do is to go to like the closest local energy minima, and hence it buckles. So and so then we have different modes of buckling which can occur depending on the constraints applied on the beam and the boundary conditions and so on. So yeah, so then the flat beam we see here is going from a stable to an unstable equilibrium as the load crosses the critical load, and hence there's an instability, mechanical instability created, which is released by buckling. So But so far we've just considered the case of a homogeneous material. But now when we look at a composite structure of two materials of different Young's modulus, which are connected to each other, we can see that we have observed that this also buckles on compression. But to just analyze how this buckles, we'll just assume the profile of buckling as a sinusoid curve, which is valid because we've seen that the different buckling modes are sinusoids. So, and one major assumption we make here is that the length of the thin layer does not change during this buckling. So to keep the length of the thin layer constant, we basically try to calculate the line integral, and the actual length of the curve made by the thin membrane. So yeah, so then we get this relation, which can be simplified to find out the amplitude of the wrinkles as a function of the wavelength of the wrinkles and the compression applied. And also there was this one very common question which asked why are the wrinkles perpendicular to the direction of compression. So as we can see, even in a simple buckling of a beam, and even when we think about buckling of just like a composite sheet of material, we can see that when you apply force in, say, like the x direction, you have the wrinkles found in the y direction. And when you extend this into the 2D, this the ridges of the wrinkle essentially extend into the plane. So yeah, that is why the direction of wrinkles are perpendicular to compression. And another example would be like when you just compress the skin of your hand, like you can just see that if you compress along the length of your hand, the wrinkles are formed perpendicular to however you're compressing it. And yeah, so then now we try to estimate what the wavelength of these wrinkles are. So yeah, so now we try to do an energy analysis. So what we're doing here is we're calculating the bending energy of the membrane, and we're calculating the energy from compressing the substrate. So here there is no energy of compression of the membrane because we're assuming that the membrane retains its length. So the bending energy of the membrane is given as a product of like the area and what is called as the flexural rigidity, which is again dependent on the Young's modulus and the dimensions of the membrane, the thickness of the membrane. And so the bending energy of the membrane is also dependent on 1 by r square. So 1 by r here is the curvature of the curvature of the wrinkle. So this curvature can be estimated by taking the double derivative of the profile we assumed. So yeah, so then 1 by r square would turn out to simply be of the order of the amplitude square divided by lambda to the power 4. And the compression energy from the substrate would again from the we're just assuming like a linear we're just assuming Hooke's law here, and as and we're trying to just we're saying that E epsilon square, this is the energy density because of elastic energy, and just multiplied by the volume of the material. So now we're trying to we try to estimate the wavelength of the material by minimizing the energy with respect to lambda. So because we know that a system always tries to minimize the energy. And so then we get an expression for the characteristic wavelength of these wrinkles, and just substituting the expression for lambda back into the expressions of UB and US, we're able to determine we're able to determine a scaling law for the total energy as well. And yeah, so now we know that there are two different modes as in the previous case. So then there's like a flat mode, and then buckling happens after a certain critical strain. So we can see that the energy of the flat mode is lower than the energy of the wrinkled wrinkled configuration below critical strain. And yeah, so then till critical strain, the system maintains flat configuration, and for like larger strains, you have wrinkles being formed. Yeah. So now we try to apply this to biological systems. So as we know, like in biological systems, the tissues grow, and now what happens when we constrict the growth is we're basically imposing compression on these tissues. So what as we so in the paper which was given, so we saw that the we saw that the the endoderm and the mesoderm layer remained remained flat, and there was no wrinkling till the E6 phase. And once you have the circular muscles which are being formed outside, so then you have a constraint in the length which is happening. And so because the length is getting constrained, there's an effective compression which is acting on the endoderm and mesoderm layers, which is leading to the formation of wrinkles. And so now as we progress in the development of the embryo, there are also longitudinal muscles being formed, and these longitudinal muscles constrain the length along this direction, which is why you also have like folding in along the longitudinal direction. So a combination of constraining length in the circular direction and the longitudinal direction gives you like the zigzag pattern. And yeah, and so this finally leads to the formation of the villi when the valleys have a relatively faster growth. And yeah, and then there's some bulging that that gradually occurs as development progresses, and you move from like a zigzag pattern to the characteristic pattern of villi. And yeah, so now we're also going to talk about what happens during differential growth. So the simplest we can we can imagine two tissues that are connected to each other but are growing at different rates. So what happens when like the top layer of the tissue grows faster than the bottom layer of the tissue is the tissue kind of again bends because of instabilities. And yeah, and because this has like a lower energy than both the tissues remaining flat, because then one would be compressed and the other would be stretched out. So yeah, so then this configuration tries to minimize the energy. And yeah, and the extent of bending here is proportional to the difference between the growth of the two layers. And so the more the difference in the growth between the two layers, the more is the bending. So and as you can see, the faster growing layer is always would always like bulge outwards, because you have just to accommodate the increased length. And yeah. So now we can intuitively try to understand why the folds in the brain happen. So what we know is that the neurons migrate from the white matter to the gray matter, which leads to like swelling of the gray matter during the development of the brain. So now we know that the gray matter is growing faster than the white matter, and so you can expect some kind of curvature to be formed in these regions. And because these again are constrained to maintain connectivity, they end up forming folds so that you can accommodate the increased growth of the gray matter region with respect to the white matter. So yeah, so even though we start with an initial condition which looks kind of smooth, we can see that as we increase gray matter, there'll be like local buckling occurring everywhere, and that's what leads to the folding of the brain. And yeah, so then there were also a lot of okay, so that concludes the discussion I had planned for explaining how instabilities cause folding in the intestine and the brain. And yeah, so now I'm trying to try to cover a lot of questions which were asked with based on why do the patterns vary between organisms, between individuals in the same species, and so on. So yeah, so in case of the brain folding patterns, so there's this study which was conducted, which basically kind of plotted the extent of gyrification of an organism's brain as a function of the brain size, which was given by the ratio of the radius of the brain to the thickness of the gray matter. And yeah, so then they calculated a couple of parameters, and yeah, and so yeah, what we can observe here is that the extent of gyrification, or like the extent of folding in the brain is dependent on even physical parameters. But the fact remains that all organisms have brains in which folding occurs. So we can see that the underlying process which leads to folding of animal brains is common across all species. And yeah, and another question was asked related to why are the structure of the villi so consistent across different species. So like the simplest answer to this would be because all these species have a common goal of maximizing nutrient absorption. So when we observe the cross section of a villi, we can see that the surface of the villi is coated with these epithelial cells, and which basically absorbs nutrients from the inside of the small intestine and passes it on to the bloodstream. So here, like this region would be the inside of the small intestine. So one way to maximize nutrient absorption would just be to like increase the number of cells. But how would you accommodate, an increasing number of epithelial cells? That would again be by increasing the internal surface of the small intestine. And yeah, so then the finger-like projections of villi basically help in increasing the internal surface area of the small intestine. So yeah, so organisms all have similar looking villi because all of them want to try to absorb more nutrients. And yeah, and there were a lot of questions based on how do patterns, whether it be like villi or like brain folding patterns, how do they change within the same species or are dissimilar and so on. So what I would like to say is that patterns of each individual is definitely extremely unique, because as Matt talked about, it's not a simple process, and there's like a variety of biological and mechanical factors which happen during morphogenesis, and it's also like a multi-step process. And because of individual's unique genetic factors and maybe environmental conditions, you have random noise being added in each step. So then there's definitely a variation of these patterns between individuals. So but again, it's also true that within the same species there are some characteristics of these patterns that um, are identical. Like one example would be the primary fold of the brain is in similar positions across all humans or across all individuals of a particular species. And yeah, and these because also patterns are dependent on physical parameters, and in a particular species the physical parameters can be expected to be similar. And yeah, so and another bunch of questions were asked related to why aren't the experiments or models capturing all the details of the biological process, or what can be done to capture all the details of the biological process and so on. What I would like to say is that the basic goal of the study was to capture the qualitative aspects of these patterns, as capturing the detail, and just by capturing the qualitative aspects of these patterns, we are able to kind of identify the major processes and understand these processes which lead to the formation of these patterns. And yeah, so the details matter, but like because the details also vary a lot, we can't really expect an experiment or a model to capture all these details accurately. And yeah, so yeah, and so this is our contact information if you guys have more questions, and I'll just take some questions right now.